Good morning, everyone. So we've covered so far immune responses to foreign things that we don't want in terms of allergy, immune responses to foreign things that we do want in terms of pathogens and vaccines. And then we've talked about immune responses to self that we don't want, which are autoimmunity, and immune responses to self that we do want. So we're going to tie this all together now with sort of immune responses to something that's sort of foreign, but it's a response that we don't typically want, and that's transplant immunology. So the idea of transplant actually goes back quite a ways. And the first real description of this, at least mythologically, is from Ganesh, the Hindu god of wisdom. And so Ganesh's mother, who was Shiva's consort, conceived of Ganesh, but didn't tell Shiva about it and sort of hid his existence. So then Shiva got home, didn't recognize him, and cut off his head. And so of course, his mom got a little mad, and so Shiva then transplanted the head of an elephant back onto Ganesh, and that's where we get the elephant-headed Hindu god. Now, it seems, looking at that seems kind of ridiculous. This is a xenotransplant, transplant from one species to another. It's not likely to work, but, I guess if you're a god, you can make that work. So the earliest studies in sort of deciphering transplant immunology actually didn't happen in humans, and it happened in tunicates. And so there were two groups, Irv Weissman at Stanford, and then David Raftos. And so there were two different kinds of tunicates. There were colonial tunicates, they form colonies. And so colonies could interact with themselves, but they couldn't interact with tunicates from other colonies. Okay, so basically that let them be able to recognize, this is my colony, that's your colony, and so there wouldn't be a cross interaction. And so this allowed them to share resources and grow better. But then David Raftos showed that solitary tunicates, which are just basically growing as individuals, could reject tissues from unrelated tunicates. Okay, so that implied that there's this recognition event. And it sounds like it's actually immune related. It is. It's because it's many of the same mechanisms that the immune system uses to differentiate you from some tissue that's being transplanted to you. Now, one of the things that David Raftos saw was that if you tried to do a secondary graft from the same donor, it got rejected faster, which implies immune memory. But the grafts from other donors were rejected at the same rate. And this was based on mouse experiments that showed the same thing. And so they mapped the genes that were mediating this self versus non-self recognition. And it mapped to the FUHC genes. We'll get to those in a minute, but they're highly polymorphic. They're the most polymorphic genes in tunicates, which allows them to differentiate between, yes, this is my colony versus some other colony that's trying to invade. But tunicates don't really have an adaptive immune system per se, in terms of how we think of it. They do have some mechanisms for it, but they're really just have immunity to foreign things. So if they don't have lymphocytes, how are the foreign cells being rejected? And it comes down to the Fester-Uncle Fester system. And so the Fester and Uncle Fester system is comprised of sort of NK-like receptor or NK receptor-like proteins. They have sort of a recognition domain, and they interact with an MHC-like protein. I know it's not drawn that way here, but it looks a whole lot like class 1. And so the Festers are proteins that are recognizing essentially MHC on tunicates. And so if it's compatible, if it's recognizing the correct MHC, then they won't reject it. But just like an NK cell recognizing a foreign cell, if it's the wrong MHC, then you don't get that not kill signal, and they'll kill it. Now, the exact mechanisms of rejection are still a little fuzzy because they don't really have NK cells, and so this is thought to be just normal cells killing other adjacent normal cells. And this really ties back into some of the earliest studies in transplantation in humans. And here we get into some sort of idea that this has been happening for a long time. And one of the earliest reports is from a guy named Sushruta in India. And he was an Ayurvedic physician who purportedly performed surgical transplant of skin. So he would reconstruct noses or ears, things that got damaged in battle. And it's not really clear if he was a real figure or if he was sort of made up. And the theory goes that he may have been an invention of the British who were colonizing at the time to gain acceptance of modern surgical techniques, saying, well, you've got this historical figure from your own country that's been doing this for a long time, so it shouldn't be any problem for us to do it. Now, that's still in a lot of debate. He actually may have been a real person, but the problem is that in the 16th century, there was a real person, Gasparo Tagliacozzi, in Italy, who would reconstruct noses and earlobes and things in soldiers who were damaged, sort of repairing their faces and other parts of their body. And he was using autologous skin grafts. And so this would be taking skin from, for example, the back and grafting it onto the nose. And if with some careful surgery, you could get that to work. But what Gasparo noticed was that if you took skin from somebody else, usually that got rejected, and so you had to use skin from the same patient. Okay. Now, where this ties into Sushruta is that many of Sushruta's manuscripts that the British were sort of touting around were plagiarized from Tagliacozzi, and so it's likely that Sushruta was sort of a made-up figure. But at least in the 16th century, so we're talking in the 1500s, before even the real discovery of America or of the North America and South America, there was people doing transplants. But whether Sushruta was real or not is not really, it's a moot point, because transplantation of skin has been done for hundreds of years. And so, but the problem was you could never get past that. So Tagliacozzi tried to do whole organ transplants. At the time, he was trying to do it for example, pigs to humans, and these were uniform failures. It just never worked. Okay, so you can't transplant between species, usually can't transplant between same individuals in a species, but you can do autologous transplants where you're transplanting skin from one site to another. And so this really started to mature into a real field in the early 1900s. Remember, this is the time of Ehrlich, and we were starting to figure out the immune system can make responses to things that are injected. But Eduardo Zirm was an ophthalmologist who was able to transplant cornea from one patient to another and restore sight to that blinded patient. Now, the only reason that this worked is because the cornea is typically an immune privileged site, so the immune system doesn't attack it. We don't give up our ability to see very easily because we would not be able to hunt and gather food. And so this worked. You could transplant cornea from one person to another. And that's still done today. Most of the vision correction surgery that you see is actually just is autologous corneal transplant, where they're cutting it off, reshaping it, and putting it back on. The first true organ transplant was between Ronald and Richard Herrick. Okay, these were identical twin brothers, and Richard developed kidney problems, and so Ronald donated a kidney to his brother. Now, this was the first successful time of human organ transplant, but it only worked because they were genetically identical. Okay, so again, this is almost like an autologous transplant where you could show that you could transplant a whole organ. For most of us, we don't have an identical twin, and so it doesn't work. But then along came Peter Medawar, who bears a striking resemblance to Mr. Bean. But he showed that you could take dizygotic cattle twins. And dizygotic means they're not identical. They're derived from two different eggs. Okay, but they're sharing a womb, and you could actually transplant organs between those cattle. And so this implied that you could have tolerance even though they're non-identical. Okay. Now Milan Hasek, who's in Western Russia, also showed this, but for chickens. And so what he did was essentially take two chicken eggs, drill a hole so they would share their share the egg yolk, and then when those chickens grew up, they also were tolerant of transplanted organs between them. But if you took another chicken that didn't grow up from that same egg pair, then it would not be accepted. Okay, so this was the beginning of showing that you could have acquired tolerance to a transplant. So today we do a lot of transplant, right? I hope many of you are organ donors. And, excuse me, this is from a few years ago. It has actually grown quite a bit. It's over 200,000 currently organ transplants per year. And that has grown every year. The vast majority of these are corneal transplants during corrective eye surgery, and then bone marrow transplants, but there are a number of solid organ transplants, including the kidney, liver, heart, etc. And the overall percentage of survival of these organs, or of the actual graft, not just the host, is about 50%. It's better for some tissues than others, but that's a pretty good survival. And so if your organs fail, somebody riding a motorcycle without a helmet can donate an organ to you, and then you likely can survive. Now, one of the tricks, though, is that transplants from a live donor work much, much better than transplants from a deceased donor. Okay, and so if you look here at the bottom, if you look at the total number of transplants for the deceased donor, there's about 15,000 transplants, and then if you look at the overall survival, the typical survival rates of them, about a third of those are surviving. But if you get a transplant from a living donor, you can see there's 3,945. With one exception, all of those survive. And that's generally thought to be because if you have a deceased donor, there's time where the tissue starts to undergo necrosis and die off. And so by the time it gets to the recipient, it's not as in good shape. Whereas if you're taking it from a live donor, there's very little time in between taking it out and putting it back into the new recipient. Okay, so it's the quicker you get an organ, the better off you are. And this has even made it into popular culture, right? There's a number of different movies that have sort of implemented this idea of transplant. And one of the silliest, right, of course, is this movie Face Off with John Travolta and Nicolas Cage, where they basically are transplanting one face to the other. And they maintain their good looks, there's no deformity. It just doesn't work, right? That's a silly example. But in 2005, there was the first face transplant. This is a partial face transplant from Isabella Dunoir in France, and she was attacked by a dog, and her face was disfigured. And then, so she actually received a face transplant from a deceased individual, and that took. Unfortunately, as the story goes, she was French and still smoked, and so that led to degradation of the transplant, and so she's had to have that redone. But this is how it evolved. I saw a recent report from this year of a face transplant, and you could really not even tell that it was a transplant. Some of the earlier examples were Dallas Wyans, who was an electrician, and when he was electrocuted, it actually burned off most of his face. And so he received a full face transplant in 2010. And that actually worked surprisingly well. I mean, if you wear sunglasses, obviously you can't restore sight and things like that, but you really wouldn't know that on the surface, that he had his entire face melted. Another example is Connie Culp, where her hopefully ex-husband shot her in the face, and essentially she was able to get her face restored by a face transplant. Okay, but this is essentially going back to tagliacosi. You can transplant skin, and if you maintain it with our current tolerogenic protocols, then you can get even from unrelated donors, you can get a face transplant. Now, I don't recommend that for anybody. Certainly does not without its consequences. So if you're thinking of changing identities, I wouldn't necessarily get a transplant. So I'm sure you've all heard of the sort of urban legend of the kidney thieves, right? Where this is where you wake up in a bathtub full of ice, and sometimes they'll write, you need to seek immediate medical attention. To make the story gruesomer, they'll usually write it in the person's blood. You sort of sit up and you feel that you've got incisions in your back and they took out your kidneys. In reality, thieves are not likely to be that nice, so they're likely just to kill you and take your kidneys. Why would they try and save your life in the end? But that has led to now the problem that if you go to some countries, if you have enough money, you can get an organ and get a transplant. And this is what it costs. And obviously, with inflation, it's probably a little bit higher now. These numbers are from 2007. But if you go to China and you want a kidney, if you've got $70,000, you can get a kidney transplant. I just wouldn't ask where that kidney came from. Okay. So it's not done in the United States, but in other countries, you can, in fact, buy organs, and it's not always clear if those organs were donated willingly. But there is an entire market for that. So let's talk about the immunology of transplantation. And so to do that, we need to clarify a couple of terms, okay? And yes, these will likely be on the exam, so please pay attention to these. As I've said, autologous transplant is when you take tissue or an organ from one individual and you give it back to that same individual. Okay, so this is you donating skin from one site in your body to another site, or them taking your cornea and chopping it up and then reimplanting it back on you. That's very common, and that usually works. Then if we go back to Robert and Richard, right, the identical twins, syngeneic is transplant between genetically identical individuals. And that's common in experimental settings where we're doing this between genetically identical mice. Okay, so it's done a lot in the lab. There's not much opportunity for that in human population because we're not typically an inbred population. And so what we have most of the time in human transplant is congeneic. And that's where you have transplant between individuals that differ at only one locus, so they're very closely related. So this would be a sibling or donating to a parent, where you have very few gene differences between one individual and the next. It's not quite syngeneic, but it's close. And so this would be things like MHC matched donor and recipients. But typically what happens is that there's not a lot of options, right? If you are in need of a kidney, you go on a waiting list and whatever comes available, that's what you get. And so most of the time, what we have is allogeneic. That means that it's transplant between two unrelated individuals in the same species. Okay, so somebody you didn't even know donated a heart. That would be an allogeneic transplant. There's two more terms, there's one more for species differences, and that's xenogeneic. Okay, so this is trying to put a pig heart into a human. That doesn't typically work just because there's too many differences. As we go down to the list, autologous, there's no differences. Syngeneic, there's pretty much no differences. Congeneic, there's one or a few. Allogeneic, there's some. And then xenogenic, there's lots of differences. And so this just goes down the list of, if it's very few differences, then your immune system thinks that's self, and so it allows it. The more differences you get, the less likely the immune system is going to let it go. Now, there's two other terms that are typically used. There's orthotopic, which is to the same site. So taking a cornea, reshaping it, putting it back on the eye, that's orthotopic. Taking out a kidney, putting it where a kidney normally should be, that's orthotopic. The other version of this is heterotopic. This is where you're taking something from its normal site and putting it to a different site. So for example, taking the skin from your back, putting it on your earlobe, that is a heterotopic transplant. This is actually seen in solid organ transplants, particularly with the pancreas, where you don't necessarily have to put it where it originally was. For a pancreas, they'll typically attach it to the liver rather than putting it over the top of the spleen where it normally is. And so heterotopic does occur. It's very common. It can be it can be from any of these, autologous all the way through xenogenic. Okay, so the evidence for this was really worked out by George Snell and others at Jackson Labs. And they went through this tremendous effort, if you remember, to show that to map the MHC genes, what they were really doing was measuring rates of transplant rejection, okay? And so the general rules are, so they made all of these inbred strains of mice. So within one strain, they're all genetically identical. And if you took skin from one mouse that had the same genetic makeup and transplanted it to another mouse, it would be tolerated. So that's essentially seen as self. But then they made some additional observations. The first is that if you transplanted skin from an unrelated mouse to a new mouse, that would typically get rejected. But just like with the tunicates, if you took this mouse then and gave it an additional skin graft from the original one, then it would be rejected even faster. Okay, so they called this second set rejection. So they started to wonder about, well, what cells are doing this? And so this was around the time of starting to define T cells and B cells. And what they saw was that if you transferred T cells from this sensitized mouse, right? So this mouse over here, transfer the T cells into a new mouse, then tried to do a skin graft, you would again get this accelerated rejection. And so this was sort of the first proof that T cells did anything, was they were mediating tissue rejection. But this sort of breaks the rules for what we've talked about, because remember, when we were talking about how T cells develop, they have to recognize self-MHC, but not recognize it too well. But tolerance sort of breaks those rules, because you get then allorecognition. And this is where you have the mouse, or whatever, pick your favorite species. The T cells are developing, but they're trained on self-MHC. And so they recognize self-MHC and then with a peptide on it. And so they're fine with that. But now they're introduced to tissues that have a different MHC on them. And in that case, then you can have recognition of the T cells will recognize the MHC, this foreign MHC, as something that's not self. So you've really broken the rules for tolerance here. Okay, so the most common ways that this is done is that the T cell receptor will just recognize the foreign MHC, and it doesn't matter what peptide's there, just recognizes that as a foreign MHC, as if that's a foreign peptide being presented on it. It's just going to recognize all it, no matter what's there. Less common is that the T cell receptor will recognize some foreign peptide that's being presented on that transplanted cell, but that confers very high avidity. Okay, and so this overcomes requirements for co-stimulation, for cytokines. There really is just, it's such a strong interaction that it activates the T cells no matter what cells are presenting it. Now, the current estimates are that you have about 10% of all of your T cells would be alloreactive for some donor. And obviously that's going to depend on the donor's MHC type, right? So if they have very similar MHC, there might be less, but in any case, you have a large number of T cells. Now, contrast that for antigen-specific cells. Antigen-specific cells are about 1 in a million. Okay, so yes, they could be reacting to something, but eventually, if that's a correct response, then it goes forward. But if it's an incorrect response, it gets shut down. The problem with alloreactivity is that there's so many. Instead of 1 in a million, you've got 1 in 10. That's 100,000 times more alloreactive cells than an antigen-specific cell. And so what happens is that you can't shut them all down. There's just too many cells, and so you get these massive responses to the engrafted tissue. So here's sort of recapitulating what I just said about, here's our normal T cell, an antigen-specific T cell. It's going to recognize antigen on self-MHC. So in that case, you would get normal recognition. You're tolerant to self-peptides because of thymus. And then if it's a foreign peptide, then you can get a normal response, okay? But if it's an alloreactive recognition, then you can have the T cell recognizing that foreign MHC with high affinity, and it doesn't matter what peptide's bound to it. It just sees that as a foreign thing. And so you get alloreactive responses. Now, less commonly, as I said, can also be because that foreign MHC is not typically recognized very well, but it's presenting some peptide which is going to trigger the T cells. So that's peptide-dependent alloreactivity. And that's much less common, but it does occur. So what's happening here, right? And at first glance, this appears to break the rules and sort of overcome all of our tolerance mechanisms, but really it's not. It's sort of the same thing that is occurring anyways. Let's say we have done a skin graft, okay? That skin graft has its own dendritic cells associated with it. Okay, so you graft the skin, you're also transferring all those dendritic cells that are in the skin. Now, during the process of transplant, if there are damp signals, which we covered in last lecture, that are associated with the surgery and sort of the damage at that tissue site, those dendritic cells will get damp signals and go to a lymph node where they again can prime T cell responses. Okay, so it's not the T cells getting primed directly in the graft, it's following the same rules. The dendritic cells are going to a lymph node, they prime the T cells will proliferate, differentiate, and then they travel back to the graft where they start killing off the graft. Okay, so this is the same thing that we've talked about all semester. There is a priming event and then an effector response. Now, as we talked about, there's this MHC dependent rejection versus peptide dependent rejection. And that peptide dependent rejection is typically called minor histocompatibility. And this is where you can have the MHCs all looks fine, it's self-MHC, but then you have other peptides that are presented that are from other genes. Okay, so here we have, if you have two identical mice, you transplant skin from one to the other, that's fine, it's tolerated. We just talked about if you transplant from unrelated mice, that graft gets rejected. But you can take mice that have the same MHC and transplant skin, and it still gets rejected, but it's rejected more slowly. And so why is it rejected more slowly? Well, again, what you're really talking about here is an antigen-specific response. So it's a foreign, essentially a foreign peptide being presented on that skin or on the dendritic cells on the skin. but the MHC is correct. And so you have about 1 in a million cells are going to recognize that new peptide, MHC complex, but they recognize it as foreign, and so they make a response to it. And so that's minor histocompatibility rejection, as opposed to they just have different MHC, right? There's about 1 in 10 cells are alloreactive, and so this is major, and this is minor. And minor, it occurs, it happens, it's just slower. So just having the same MHC is not enough. If you have other differences in other genes resulting in presentation of other peptides, then you also can have rejection. And that's sort of shown here. Here's our donor, right? It's got some self-protein. We all have small nucleotide polymorphisms that result in amino acid changes because we all have mutations. So those are then processed, just like we've talked about. They're sent through the transporter of antigen processing and presentation. They bind to MHC and they make it out to the cell surface. Many of the epitopes presented, they're identical between the donor and recipient, right? So those are not a problem. The problem is if the recipient has T cells that are tolerized to this peptide, and now you have changed the amino acid sequence of that protein from the donor, now you're presenting a different epitope. And so the T cells will come in, they'll see this antigen and think, well, that doesn't look like me, so I'm going to make a response to it. Okay, so those are the minor histocompatibility antigens. Okay, so there are a number of different ways that this works, right? Major histocompatibility antigens are just this TCR dominant recognition where it doesn't care what peptide's there. Now, typically the responses are primed by professional antigen presenting cells. In some cases, it's thought that it could be other cells, but a lot of that is because that's a very high affinity interaction, and so you overcome a lot of the requirements for co-stimulation. Well, if you think about it, that's one of the ways that you maintain tolerance, right? If a dendritic cell is presenting a self-antigen and you have a T cell that can recognize that, if it doesn't have co-stimulation, if it's not expressing the co-stimulatory proteins, that T cell becomes anergic or dies off. But if you have this high affinity interaction, because it's a foreign MHC, then that overcomes some of those co-stimulation requirements and breaks tolerance. Now, for minor histocompatibility antigens, oftentimes it is if the recipient and donor have the same MHC, then you're just expressing these and presenting them. But many times, if you can have donor proteins that are being secreted, and then these are taken up by cross-presentation in recipient dendritic cells, and then you get a response to that. Okay, so it can also be not just intracellular proteins, it can be surface proteins or secreted proteins that can initiate this response because of cross-presentation. Okay, so how do we test this? Well, there's really two ways to do this, and it's really looking at the T cell responses, okay? Now, for CD4 cells and CD8 cells, we measure this differently. And I'm sort of presenting the older versions of these that use nucleoside, radioisotope labeled nucleotides. And so the first one is you'll take your self lymphocytes and irradiated donor cells. And so the donor cells cannot proliferate because they've been irradiated, and you mix those together. And what you're testing for is, am I going to get a strong response? Is there going to be a lot of alloreactive responses? So for CD4 T cells, mainly we measure proliferation, okay? And so you put into that culture irradiated or radioactive thymidine. So this has a hydrogen, radioactive hydrogen label on it. And as that's incorporated into the new DNA, then those cells become radioactive and you can measure that. For CD8 T cells, we measure it a little differently. You label your target cells with radioactive chromium, which is taken up by the cells. And then if the T cells react to that, then they'll actually kill those cells and you get release of chromium. And so you're measuring how much of chromium is released. That tells you for CD8 cells how much alloreactivity there is. And so this is often done prior to transplant. If you want to get a sense of how well the organ or tissue is going to be engrafted, you'll do a mixed lymphocyte reaction to see how much alloreactivity is there. If it's MHC differences, typically it's very strong. You get lots of proliferation of CD4 cells, lots of killing by CD8 cells, and so you don't want to do that organ transplant. For minor histocompatibility antigens where they share the same MHC, it's just some different peptides are being presented, then you typically get very low mixed lymphocyte reactions because there's just far fewer cells that can do that. Okay, so here's some general rules for transplantation. If it's an autologous, or in this case, syngeneic transplant, where you're taking tissue or skin from a mouse and transplanting that to the same strain of the mouse that has the same MHC, that is autologous or syngeneic, that is typically accepted. Okay, so that works. But if you instead take skin from unrelated donors and transplant it to different recipients, so here they have different MHC and they also have different background other genes. So you're going to get both major histocompatibility or major antigen rejection and minor antigen rejection. In that case, the major is dominant. And so you'll get fast tumor rejection. Okay, so it doesn't work. But on the other hand, if you take mice that have a different background but share the same MHC, then you only get the minor histocompatibility mismatch. And so the tissue is typically still rejected, but it's a slow rejection. It takes weeks or months for that to happen. So that slower rejection can be managed with things like immune suppressive drugs that will inhibit the immune system and allow you to get organ transplant to work. So I realized that when I posted these slides, that I included the answers to them. So we'll just go through these together. But the question is, if I take tissue from a parent and transplant that to the offspring, so I've bred together two unrelated strains of mice, and I take the parent tissue and transplant it to the recipient. Okay, so the parent has BLAK6 background, the offspring has BLAK6 and C3H background. The parent has H2D, where the recipient has H2D and K. And you ask, will the graft be rejected? The reverse of that is to take tissue from the offspring and transplant to the parent. Okay, so which of those will work? Well, parents can donate to their offsprings because the offspring has BLAK6 antigens, so it's tolerant of that. It also has H2D, so it's tolerant of that. And so it's essentially seeing it as a version of its own self. Okay, so the minor antigens are shared and the major antigens are shared, the MHC are shared. But it doesn't work the other way around. Okay, so when you look at transplant between the offspring to the parent has BLAK6 background, but the donor has these foreign things, right? Peptides from the C3H background. And so that the parent automatically can see that as different. Additionally, the offspring has on its cells, it has H2K. Well, the parent has never seen H2K before. It only has H2D. And so you have both MHC differences, so those are the major antigens, as well as the background differences. So in this case, C3H background proteins. Okay, so this doesn't work that way around. Now, some of the other questions, we'll go through these in a second. So if you transplant between two identical mice from the same strain, if it's a female to male, or sorry, it's female to female, that works, right? They have the same antigens. If you do it male to male, that also works. The question is, what about female to male or vice versa? So female to male typically works. Okay, so they're transplanting from female mice to male mice. That typically works, but it doesn't work the other way around. Transplanting from male mice to female mice, typically you get rejection. And the main reason for this, one of the main reasons, is that some of the proteins from the Y chromosome are very immunogenic. And so you get presentation of those as minor histocompatibility antigens, and you get eventual graft rejection. Okay, so whenever you're thinking about will a tissue be accepted or rejected, you have to consider, is the MHC the same, right? Does the recipient, is the recipient trained on that MHC? Have they seen it before? And secondly, are the peptides that are going to be presented on that MHC the same or different? So which of course brings up the question of, well, why do mothers not reject the fetus, right? The fetus has, for example, if the fetus is a boy, it's got Y antigens. Additionally, the fetus has got half of its MHC from the father. The mother hasn't been trained to do that, or the mother's immune system hasn't been trained to be tolerant of that. So in general, we would expect the fetus to be rejected. And that does actually occur sometimes. We've talked about some examples of that. So there is, but the way that this is maintained is that in general, there's a placental tissue barrier where cells from the mother are not interacting with cells from the fetus. Okay, and there's mainly blocking T cell access to the developing fetus. Now, at that interface, there's a number of things that are happening. There is expression of indoleamine deoxygenase, which is perhaps the more best well-known pathway. That is, it's being expressed extracellularly, and it degrades tryptophan. Well, tryptophan is a limiting amino acid. It's very present at very low levels, and so if you have an IDO, then you don't have tryptophan, so you can't, those cells can't make proteins and they die off. Okay. Usually, but there is transfer of antibodies between the mother and fetus, and so there's usually not enough natural ABO antibodies to cause disease. We talked about erythroblastosis fetalis. That's where there was a priming event from the first child. The mother develops high level of ABO antibodies against the second fetus. And so you can block this with ROGAM, as we talked about, but you can get rejection from antibodies, but the T cells are generally have blocked access to the developing fetus. So how does antibody rejection work? Well, you can have antibodies, either pre-existing natural antibodies that sort of develop from this strange B1 B cell subset. And these allow you to make low-level responses to incoming microbes. But if you have lots of these, for example, ABO-specific antibodies, then what will happen is those will bind to the donor cells. Here's a donor cell. And then they will activate the complement cascade, right? So there's C4 and C2 coming in. Then that acts as a C3 convertase. That gets deposited. Eventually, that acts as a C5 convertase. And then you get activation of 6, 7, 8, 9. All right, the same pathway that we've talked about before, but that is the membrane attack complex. And so the cell loses the membrane potential and dies. Okay, so most antibody-mediated transplant rejection is complement-mediated. Other examples, it can be NK cell-mediated or other innate cells that have these antibody receptors, but the main one that I want you to really look at is complement. Now, what are those antibodies seeing? Well, they have to see something as foreign. So it can be blood group antigens, right? Those are differences in glycosylation. Natural antibodies typically recognize glycoproteins or glycolipids. But it can also be, if it's a foreign MHC, you can have antibodies recognizing the foreign MHC, can be tissue-specific antigens that become exposed. Anything that you have antibodies that are recognizing something foreign, you can get antibody-mediated rejection. But they can also do something that's called hyperacute rejection. And this occurs mainly when you get a second transplant. So an example of this is maternal antibodies to fetal ABO antigens. But it can also be if you've gotten a kidney and then you try to get another kidney after that one fails, and then you have accelerated rejection because you have a lot of antibodies against it. Okay, so here you have a normal antibody or a person that gets a second kidney graft, and you have lots of antibodies. And what happens is that these antibodies then will respond very quickly, and they will actually cause destruction of the endothelium, complement mediated or FC receptor mediated destruction of endothelial cells of the vasculature. And now you just don't have any blood flow to the organ, and so that organ dies off very quickly. And usually they show it in purple because it becomes hypoxic. But basically, if you have hyperacute rejection, you're just shutting down the vasculature, so there's no blood. Excuse me.
Dr. Blackman?
Yes.
Does this not occur if you get a transplant from a different donor?
It actually depends on how closely the two donors were related. So for example, if you were to get a second kidney from the sibling of the first donor, then yes, this is very likely to occur. If they're totally unrelated donors, it may occur, but it's less likely to happen. But for example, when people have tried to do organ transplants from other species like pigs, if you try and do a second one, it gets rejected in this hyperacute way. Okay, so how do we deal with it? Well, you've probably all have heard of immunosuppression, right? Where these are drugs that block T cell proliferation or function. This has most notably been in the news recently because most of the COVID symptoms are T cell mediated. And so if you block that with corticosteroids, then you can block the T cells and get less disease. It's not yet clear if that impedes clearance of it, but of the virus, but it's also used very commonly in transplant. So for example, if you get a kidney transplant from an unrelated donor, in order to prevent the T cell response to that, you go on lifelong immunosuppression. Okay, and so this is the survival rates. So if you have no differences in MHC, so your MHC matched donor and recipient, you get a fairly high survival rate. It's around 50%. But if you then put that patient, the recipient, on cyclosporin, which is going to block, in this case, blocks mainly CD4 responses, but because they control everything else, again, that's the ones that you want to inhibit, you get much better survival. Okay, so you go from here to here. This can also work when you have non-MHC matched donors. So here you have 3 MHC differences. Cyclosporin can take you from about 25% up into near 40%. If you have a completely MHC mismatched donor with 6 MHC are different, you still get a roughly 10% improvement in survival. But again, the problem is that the more MHC differences you have between the donor and the recipient, the more alloreactive cells you'll have. And so if you have lots of MHC differences, it becomes very difficult to block all of those, even with high levels of steroids or cyclosporins. The other problem with this is that now you're globally suppressing the immune system. So if you've ever been around somebody who is on organ transplant, they wear a face mask, they limit interaction with people who are sick or displaying symptoms because they're immunosuppressed. They cannot respond to virtually any infection coming in. Okay, now one of the sort of variations from transplant is bone marrow transplant. And this is a little different because what you're doing is transplanting cells that give rise to the immune system. Okay, but this is often done in cancer patients, right? Who they're giving chemotherapy with anti-cancer drugs, or they're receiving radiation. What that mainly does is kill off any rapidly dividing cell. Well, that's great because you're killing off the cancer, but you're also killing off the developing immune cells. Okay, so those are rapidly dividing and differentiating. So you have to replace those immune cells. And the way you do that is you take bone marrow from a healthy donor. In adults, right, your bone marrow is mostly in your flat bones, in your hip, your sternum. And that is, so if you go to donate blood, they'll take a long needle and inject it into your hip. They'll take out those healthy bone marrow cells and put those into the recipient. And that restores hematopoiesis in the recipient, giving rise to all of these new immune cells. But there's some problems with this, right? If you're taking from MHC mismatched recipient and donor, then you have this problem of, well, are those cells, remember, those cells are going to be trained on self MHC and peptide. And the question is, will they attack the recipient? And the short answer is yes. We talked about this a little bit with immunity to tumors, where you want graft versus tumor effects. Okay, so you're taking the bone marrow and transplanting it, but in that bone marrow, you have lots of other cells besides progenitor cells. You have T cells and innate cells. Okay, and so those T cells that are transferred along with the bone marrow will then be able to go out, and if you're a cancer patient, sure, great, they can attack the cancer. But they can also attack other immune cells. They can attack tissues from other organs, and that causes graft versus host disease. Okay, that's bad. Graft versus tumor, in this case, graft versus leukemia, is attacking a cancer. That's what you want. But the cells attacking the host is a bad thing. Now, one of the solutions to this has been to, okay, before we transfer the bone marrow, let's just take out all the T cells. Well, that kind of works, but then it doesn't work in the long term because then you have other problems we'll get to in a second. Okay, so graft versus host disease really is when the T cells in the bone marrow that you're transplanting are attacking host tissues. And so here's a response to the skin. You can see that it can be quite severe. Graft versus host disease can kill you in a matter of days, right? It's very severe. You have to manage it with high levels of steroids to suppress the immune system. Now, a lot of the response of graft versus host disease ends up being in the mucosal epithelia, primarily in the gut. And a lot of that is because there's additional layers where the irradiation damages the mucosal epithelia, and you get lots of microbial products that are being transferred across the epithelial border. Those are providing PAMP signals, so you get lots and lots of T cell activation. So what I want you to know from this is that bone marrow transplant has this additional problem. It's not just the bone marrow progenitor cells you're transferring, it is the T cells. So you have to deal with that a little separately. Okay, but in a cancer patient, you really want to say, okay, well, what's the best way to do this? And so if you look at cancer patients, if they're syngeneic or from an identical sibling or an identical twin, so autologous or syngeneic, you get very low graft versus host disease, right? On the y-axis is our graft versus host disease, but on the other hand, there's no graft versus tumor effect. And so bone marrow transplant from genetically identical individuals doesn't really work very well. If you have T cell depleted bone marrow, that also doesn't work very well. Why? Because you're not transferring T cells that can mediate that graft versus tumor or graft versus leukemia effect. Now, if you transplant between bone marrow from an HLA or MHC identical donor who is highly related, that does work a little bit better, but you still get some incidence of relapse, and you do get some graft versus host disease. Okay, so it's sort of in the middle. Why? Which kind of antigens are there? Well, it's not the MHC, so it's not MHC alloreactive, it's minor histocompatibility reactive. So you eventually do get GVHD and graft versus tumor effects. So you always want some of the other. Most of the time now, most cancers, we're now doing bone marrow transplant from unrelated donors. So these are MHC mismatched. So the T cells can recognize the donor, can recognize the recipient's MHC. T cell from the donor in the bone marrow can recognize the recipient's MHC. And so you cause both graft versus host disease, but they're also causing graft versus tumor effects. And so you get high probability of disease, but you get very low relapse because they're killing the tumor at the same time. So how do you manage this? Obviously, graft versus host disease can kill you. And so we use the same things that we used in autoimmunity, where we're using immunosuppressive drugs. So for example, the one we just talked about, cyclosporin, it inhibits the production of cytokines, primarily, excuse me, primarily is blocking the production of IL-2 from CD4 cells. And so if you don't have T cell growth factor, the T cells don't expand, and so you can sort of trim the response down. You can also use methotrexate. This will block DNA and RNA synthesis. And so if the T cells can't proliferate, they can't synthesize new DNA, you can shut down the response. And you can also use steroids, okay? And those are mainly going to affect the same thing as cyclosporins, where you are shutting down expression of things like, T cell growth factor. So what typically a physician will do is do an allogeneic transplant. So you get the best graft versus tumor effect. And then if you see graft versus host disease, then you start to give them doses of these immune inhibitor drugs. Now, based on this, what was done and this was pioneered by E. Donald Thomas and others at the Fred Hutchinson Cancer Center, was to say, okay, well, if we take mice and we do a skin graft, and then we give a skin graft again, we get a hyperacute rejection or accelerated rejection, what we call the second set. Okay. But if we block the CD4 T cells, okay, and we'll still get rejection. Sorry, let me step back here. If we block the CD4 T cells during that first graft, you'll get graft acceptance, and then you do another graft, and you get continued survival of both grafts. Okay, so what this showed, you can sort of ignore the middle lane. This is just where they waited until the CD4 antibody went away. But if you just take the left and right columns, that indicates that if you control the CD4 cells, then you can get CD8 activation, but just like in what we described in tolerance, without CD4 help, CD8 cells will eventually die off and go away. Okay, and this worked really well in mice. So they tried to do this in humans, and it resulted in even worse graft versus host disease. Okay, now the mechanism is still not really known, but what's thought is what you're doing is depleting regulatory T cells. And so in humans, if those are much more important, you take all the regulatory T cells out, well, now there's nothing to shut down the response. And so in humans, this did not work at all. This was a spectacular failure. And so what happens, this comes all the way back to allergens. How do you desensitize against allergens? Well, you give increasing amounts of that antigen in a non-inflammatory situation to try and get more and more regulatory cells. So here's our donor. We're putting in hematopoietic stem cells, and as those go in, they will then be recognized by the recipient cells, or they will respond to the recipient. Okay, but on the other hand, what happens over time is that the recipient starts to generate more and more regulatory cells against those foreign tissues. So even if it's a completely mismatched donor and recipient, the recipient eventually becomes tolerant of the donor if you can suppress the immune system so they then don't die. So this is called mixed chimerism, where even though they have this foreign MHC on the target on the allograft, it still can cause tolerance to it. The way this is shown is to basically, um, if you take recipient cells, mix them with donor cells in conditions where you get lots of regulatory T cells. In this case, they're culturing them ex vivo in the conditions that favor regulatory cell development. You put in those regulatory cells with bone marrow from the donor into the tolerizing recipient, then the graft is not rejected. These regulatory cells are going to block any T cell function that's coming in. Okay, so this is a better approach and has worked in humans. You still have to moderate this with immunosuppressive drugs, but as long as you can get the immunosuppression to work long enough, eventually you can get tolerance to the donor tissue. But there's another problem, and this one was not really recognized as something that happens a lot, but the problem is that you can also have, if you have infected organs, so for example, you're transplanting a liver from somebody who has a viral infection. Now, we typically don't think of this, but this actually happened. And so there's a virus that I work with called lymphocytic choriomeningitis virus. We've talked about that somewhat in class, that it's a virus that doesn't really do much. It has 4 genes, and so it's not really um, pathogenic in humans per se. But what happened was in 2003, there was somebody had bought a hamster from a commercial pet store. That hamster had not been screened for LCMV because it's just not typically done. And so that person who bought the hamster got infected with LCMV. They then went out, crashed their motorcycle, and became an organ donor. And all of the 6 recipients of organs from that patient, from that donor, died. And people really didn't know why did they die. And what turned out is that it was basically you're transplanting an organ. There's such a high level of virus that it spreads throughout the body, and you're dying from the immune response to the virus. And so that was in 2003. There was an additional episode in 2003. This is in the United States, where all 4 of the recipients died. In 2005, they started to recognize this, and one of the 4 recipients survived for a while, but eventually did die from complications. 2008, 2008 in Australia, and 2008 in the US, there was another 5. And it wasn't really until we discovered, well, we could do broad-scale immunosuppression and antiviral treatment, we could actually get some of these patients to survive. Now, in many of these clusters, they could show that the donor had recent contact with a virus-infected hamster or other rodent. Okay, and they were looking at this. They would go back to the donor's house, and they had just bought a hamster. Hamsters are severe carriers for LCMV. But this isn't the only virus. This happens in organ donors from CMV positive donors to CMV negative donors. And so you're transplanting a lot of virus. And so you get an immune response to it. But because you're transplanting so much, the immune system can't catch up and it will kill you. Essentially, you get hemorrhagic disease. So this isn't necessary transplant rejection, but it still is a very serious problem in transplant. Okay, so I just want to cover a few things about the different lectures at the end of this semester. And we're going to group these together, right? Vaccination or infection, you want an immune response to this. So it's a beneficial response. It is injection of, or exposure to a foreign antigen, and there's some adjuvant there. There's either PAMPs, right? Or you're including something in the vaccine that allows the innate system to get activated. Now, there's not really genetic predisposition to this. There's a little bit, but mainly there's just variation in how well you respond. So when you're talking about this, I want you to think of what are the innate signals? Well, it's the PAMP receptor signals, right? So if it's a Haemophilus influenza vaccine, you're going to have things that are going to activate toll-like receptors. If it's a viral vaccine, you're going to activate Rig-like receptors. So it's really a PAMP receptor. How did you initiate a B cell response? Well, it's antigen and IC3B complexes that are being transported by macrophages and delivered to follicular dendritic cells. How did you get T cell activation? Well, that's dendritic cell activation. So either at the site of injection or dendritic cells within the lymph node, and they're going to present this to T cells. And that can be either cross or direct presentation. What are the downsides to this? Well, if you're unable to control the vaccine, that could be damage. You can have too much of a response, or for vaccination, you can skew the response in the same way. We've covered all of those things, but I'm trying to relate all of these together so that you get a sense that it's actually the same rules in each situation. So autoimmunity is a bad response to self-antigen. Now, there's an initiation, just like for vaccination infection, there's exposure, right? So there has to be an initiation event. Well, it's probably, for many autoimmune diseases, we can show it is an infection or there's some damage, okay? And then eventually you get either cross-reactive, the two main mechanisms we talked about are cross-reactive to self-antigens, that's molecular mimicry, or as you get damage to those tissues, you release antigens and then you get the epitope spreading. Now, this does have a genetic predisposition where you have differences in tolerance mechanisms, then you can have severe predisposition, or particularly MHC alleles are going to present both a foreign peptide and a self-peptide, and it looks very much the same. So what are the innate signals? Well, if it's an infection, you can have PAMP receptors. If it's epitopes, and that's the molecular mimicry or cross-reactivity. But if you're getting lots of damage and epitope spreading, then it's the DAMP receptors. But the other mechanisms are exactly the same. For B cells, you need antigen IC3B complexes on follicular dendritic cells. You're going to have dendritic cells presenting to T cells in the lymph node. There's some variations, but it's really the same mechanisms. Okay, now what happens? Well, essentially, you're getting killing of your self cells. So it's still controlled by CD4 cells and it's mediated by CD8 cells or antibodies, but that's the same problem that you have in a viral infection, where too much of a response is going to be detrimental. Now, allergy is a bad response to a foreign thing. Okay, so the initiation, again, you have to have some exposure event. And so this is somehow there's getting exposure. Many of the allergens we talked about are good at getting past epithelial barriers. There is a genetic predisposition, right? If you have certain MHC alleles, you're more likely, females are more likely to get allergies than males. There's a number of different genetic things that will clear that. For the innate signals, we're not really clear what happens there. It can be damage. It can be that you have an infection going on at the same time and then you develop allergies, but it's not really clear what the innate signals are. Or the antigen delivery to the B cells, There's free diffusion because these are typically very small proteins, and so they can go through the follicular reticular network. But again, they have to be displayed on follicular dendritic cells so the B cells can recognize them and initiate an antibody response. The dendritic cells are going to pick up the antigen, travel to lymph nodes, and present to CD4 cells. I do want you to know what are the different types of hypersensitivities, right? And they really break down to, is it mast cells in IgE, is it complement in IgG, or is it mast cells in IgG, okay? Or is it T cell mediated? So you should, for the exam, please become familiar with those 4 different groups. Okay, then we come back to tumors. That's a good response to self. And so the initiation mainly is the hypoxia. You can think of that as really generating DAMP receptor signals. Okay, and remember, the hypoxia makes those cells initially susceptible to NKT cells that activate NK cells, they kill them, and then you release the DAMPs. Okay, so it's the same, you have to have an initiation, so you get exposure to a tumor, essentially the tumor develops, and then the NKT cells will start activating things, you get release of parts of that that are seen as foreign or seen as something that shouldn't be there. Those are the DAMPs. There is some predisposition, right? And mostly, but this is again, cancer predisposition is primarily different than the immune system because it's how predisposed are you to getting cancer? Do you have mutations in HER2 that are going to be predisposing you to breast cancer? Those are different than predisposition of the immune system. Now, you don't really have much of a B cell response to tumors, but typically it's thought the things that are being released from the cells are being taken up into the lymph nodes, being presented on follicular dendritic cells, and again, you get B cell response. For dendritic cells, they're also picking up stuff that's being released. They go to a lymph node, and then they're going to activate T cells. So it's the same mechanisms. And these are sort of cheat sheets for you for the exam to differentiate between these different responses. Now, what happens, right? You get CTL responses to the tumor. That's a good thing. But then you should also know, well, that can be a bad thing if you're doing a bone marrow transplant and you want the graft versus host disease, or sorry, graft versus tumor effect, but not graft versus host disease. Then finally, transplant is a harmful response to an introduced semi-foreign cells or semi-foreign antigen. That really requires an initiation event. There's minimal genetic predisposition, but it can happen if you are predisposed to having a very strong responses to things, you're more likely to have transplant responses. But again, what is the innate signals? Well, it's DAMP receptors. As they are activated, these are being released from cells, and then the tissues, those cells are releasing antigens that are then mostly freely diffusing to the lymph nodes and binding to follicular dendritic cells. Dendritic cells are going to pick up those antigens and they're going to activate T cells. Okay, so transplant mainly is damage to the transplanted organ. But again, if it's bone marrow transplant, you get graft versus host disease because of the T cells coming in that bone marrow transplant. And that can help in graft versus tumor effects, but in a transplant, you typically don't want that. Okay, so I'll end there and take questions.
Dr. Botman, just looking at the syllabus, do we have class on Tuesday?
I think we do.
Okay. It just wasn't there on the syllabus.
Let me pull that up right now. Huh. Well, it is on the syllabus. It's just it's shifted down below Thanksgiving. Yes, we do have class on Tuesday.
And that's the last lecture that's included on the on the exam, right?
That's correct. Okay, if there's no other questions, we'll see you on Tuesday.